objectives, and to help you, and to help me also to organize my lectures. So there, I mean, the question is, what are these? These are the identify the pathways that either convert sugars and generate sugar nucleotide for glycan biosynthesis, recognize glycogens, glycoconjugates at the component of the cell surface, component of the cellular matrix, component of bioinformatics, and recognition markers. All of those things are present in our system. Now, whenever we get something, we're looking for the bioinformatics to give some explanation why it was resolved inside the body is happening all the time. So we have a lot of science going on in the human body. And they analyze the glycosaminoglycans are unbranched polysaccharides, which contains a uronic acid, or the glutyronic acid, or iuronic acid, which is an isomer of each, and then amino sugars called hexosamines. It could be glucosamine, or it could be galactosamine, and they could be acetylated, they could be sulfated, so the name chain would change. Then this, then we'll have when this unbranched polysaccharides contains this one, which is repeating disaccharide units, those will be generated of the, they will be named as the glycosaminoglycans. And when the glycosaminoglycans attach to the protein core, that becomes proteoglycans. And they have a very high molecular weight. They have high molecular weight macromolecules. And hyaluronic acid is one of them, which is purely glycosaminoglycan. There's no protein associated with that. But it will do interact with the other molecules of the same class. There's no covalent interactions. It's mostly physical interactions because the compounds. Now what we're dealing with is not only the neutral molecules, but they will be highly charged, polyanion, negatively charged molecules. And so they will. One of the things that they will not aggregate because of have the same charge. There will be what? Repulsive. But at the same time, because of the proteoglycans and this on the protein part will have a different charge, and these are negatively charged, and positively charged, they can interact in certain ways. And they will recognize that the glycosylated proteins may contain ending oligosaccharides or holding oligosaccharides, depending upon which amino acid is linking up. There's asparagine residue, or the serine residues, and there is a microhydrogen that exists. And then the glycans play an important role in cellular or molecular or macromolecular recognition. Because when the cell-cell interact, all the cell signaling starts, all the receptors in the cells, they are all glycoproteins. And the glycosaminoglycans, because they are so huge and in water, it forms like a gel-like structures. And then they'll interact with the other protein molecules, gives you the extracellular matrix. Sometimes when you're talking about our oral cavity, which is the saliva, has a lot of those proteins. The mucin is one of them. The asparagine-linked glycoprotein is one of them. And they are synthesis and secretion connected by what? Neuronal interactions will be there. And then you have a different gland, the parotid, what other glands you have? Sublingual. Submandibular. So those are the ones who will be responsible and continuously secreting those molecules. It's not, saliva is not water. Water is one part of that. So saliva is viscous. So because of the nature of the component present in the water. So also then you identify the glycoconjugates, which is the uronic acid and neuraminic acid. Then biosynthesis of glycans as a diverse function, and recognize the asparagine-linked glycoproteins as a co-translational event, and how they're being built intercellularly, and then they have a different role to play. Either they will, all the proteins, probably normal proteins, they can stay inside, but when they're glycosylated, many proteins have to get out, the secretory proteins, because that's what their function is. Some of them will be made up of the membrane, a component of the membranes of the recognition marker we're talking about. Some of the protein, some of the enzymes, which are glycoproteins, needs to be targeted to lysosomes. If you know the cell biology, because lysosome has a highly acidic pH, and they have glycosidases, they break down all the sugars residue and to degrade. They're basically protecting us from any kind of infections and other things. So they're, and those have to be, the mannose has to be phosphorylated in the 6th position, and there's a receptor. It goes in there, and then it delivers its cargo, a receptor recycles back, and so forth. So that's what the kind of molecular markers or recognition markers to build there. It's not just it's not a linear fashion. That's what you said. Okay. So there are some blueprints, some signatures, and has to be followed. All right. Let's see how we can swell. And then obviously, if you have any problem associated with that, then you can have, I mentioned about this, as a passing comment, you can have a CDG, which is congenital disorders of glycosylation. I think there are about 70 such cases have been identified as very rare genetic disorders. And it's global, it's not necessarily in one particular community or in a particular place. I think the majority of the cases first was identified maybe in some part in Europe. And then since people migrate, so we also get to see them here as well. So the other was the blood group substances. They are made of your one single sugar will tell you what blood group you belong to. So that's a very important aspect. And I mentioned over the last story, then they have other, and heparin, which is it's a glycosaminoglycan. It's a heteropolysaccharide. That's heparin. What is the role of heparin? It's an anticoagulant. So, and then we have diseases related to mucopolysaccharide, saccharidosis. The initial name was given the glycosaminoglycans. The earlier name was given mucopolysaccharide related to mucostat molecules. Muco means it's sugar. And then you have other deficiencies leads to other pathological conditions. But let's look at the structural component. So let me, if the time permits, I'll do the proteoglycans and glycoproteins. Obviously, I'll touch barely the peptidoglycan, but I will not do that too. Glycolipid, I'm not touching that. So here the peptidoglycan is basically, which is the cell wall, the bacterial cell wall. The first one is the gram-positive bacteria has the first layer is the peptidoglycan. Gram-negative is somewhere is hidden. So that's why you remember that we have a different antibiotics to attach to kill this bacteria if you have a different of the infection. So if you have ones like a penicillin and others, well, you try to do it, probably will go with the gram-positive bacteria that are not the gram-negative, right? I have to go to because of they have this peptidoglycan code. Now the question is, what is the peptidoglycan is? Here's the information that is the gram-positive bacteria. Here's the peptidoglycan at the top. So you can very easily accessible. And gram-negative bacteria is between the two lipid bilayer. This is the outer layer, is the inner layer, that's what is included. So your therapeutic cannot have filtered through down all the way if you try to use this one. But they are highly modulated, what's it called, the lipopolysaccharides. So here's the huge, here's a lipopolysaccharide, and the subunit of bacteria, you see that. Now here's the bacterial cell wall. It will make try to make it a little bit easier for us to understand that what exactly it means. These are disaccharides units by the two different sugars, the two color coded, green and red, right? Each one has its name. This is the N-acetylmuramic acid. It's a 9-carbon sugar, remember top, and the other one is N-acetylglucosamine. And they are being repeated X number of times. And so now you can see the 1, 2, 3, those tanks are being made. But if you keep them, they are not going to be strong enough, right? You have to make them stronger. And so that's what you have. Then each one of them will have attached to this 4 amino acids. The amino acids are L-alanine, D-gluconic acid, L-alanine, and lysine and D-alanine as well. Okay, so you have the sugars, and then those 4 amino acids are hanging. Like you're thinking about that your 2 earrings are hanging there. But okay, that's good for them, but in this case you have to stabilize so they don't move. So you have to do some sort of a cross linking. And the cross linking is done by this anti-lysine link. So that makes the structure is extremely rigid. You cannot break it. You can break it if you have the proper enzyme to break it. You can break that. So this bond is again it's a glycosidic linkage, and each one of them will also have a reducing end. The other components, as I mentioned, the beta-1,4 link, and the cleavage side, say for example here, you can break the bond here. Who does it? Which enzyme? Where can we find lysozyme?
Tears and saliva.
And again?
Tears and
In
saliva.
the tears, right? So that's why we have very, very, very little eye infection. Now you know why we don't get eye infections so frequently, because we have this, bacteria is everywhere. We try to colonize it, but we'll not be able to do it because we have a self-made antibiotic kind of thing is there, right? Biologically active antibiotic, and we break it and we take it, make a soluble molecule, and we come out and do it. Okay, so this is just the one segment of the Staph aureus. It's a very nasty bacteria. You know that the Staph aureus infection is very bad, and most of the time in the hospital area, they are there. They have to deal with that. Some people come down with the infection. Okay, so we mentioned this. So the proteoglycans is here. It's a class of complex macromolecules, which is 95% carbohydrate and polyanion. Okay, so it's been negatively charged molecule. The carbohydrate chains called the glycosaminoglycans, and RDA is called glucopolysaccharides. They are linked covalent to GAG when they're linked to protein core, which is a very minimum percentage of protein. So then we'll be calling the proteoglycans. So there are 6 distinct classes of the GAGs are recognized at this moment. So there are long unbranched heteropolysaccharides. They're made up of 2 sugars, disaccharide unit, repeating disaccharide units. It repeats X number of times. The length obviously varies, and some or other, somebody controls the length. You don't know who does it, but it does. And the terminate. So one of them will be hexosamine. I mentioned N-acetylglucosamine or N-acetylgalactosamine, and the other one will be uronic acid. It could be either glucuronic acid or its isomer from hydrochloric acid. The other common substitutes are the sulfate. It could be O-sulfations, the hydroxyl group could be sulfated, or the amino group could be sulfated. All the sulfates are negatively charged, so that means they are adding uronic acid. The carboxylic group is adding negative charge, and this one adding more. So that's why it's called the highly or polyanion. So it has more negative charge to that. And that's already mentioned. The exception, only exception is in this class is the hyaluronic acid. It's 100%. It's a glycosaminoglycan. It has never been attached to the protein. And then they have electrical charge and macromolecular structure. Therefore, they will have a tremendous amount role to play in biological roles. This could be as a lubricant. It could be like as shock absorbers and all of those. It does. All our joints. They have this fluid. These are the proteoglycans and glycosaminoglycans. So that makes our joints mobile. If it dries up, because we are drying up of those fluids. And I don't know how to get them back at this time. But I know the people have been working on it. And then some of them, so thinking about your car, right? The same way that you need a lubricant and also you need a shock absorber so that when you're driving, they're not going up and down the bumpy ride. So you have to maintain those. So they are providing the kind of a cushion so the cell can function the way it's supposed to. And then this is very general way to present the proteoglycan. This is the segment you have to hear. This is the glycosaminoglycan. And this is the protein part. So carboxy terminal, the N-terminal, there's a glycine, X-glycine. And this ring, hydroxy group, the OH, hydroxy amino acid has to be. The first 3 sugars have to be attached. This is called the linkage region. And the rest of them, they're repeating units of the disaccharide. And here an example is given the chondroitin sulfate, because the chondroitin sulfate, we have the N-acetylgalactosamine and glucuronic acid, and it's 4 sulfate, 4 position of the galactosamine sulfate, and repeating n number of times. And this extra glucuronic acid is here, but this is the one which is repeating n number of times. And this is a linkage region. The whole thing, if you do a hydrolysis, you take this part and the protein part comes out and you get the glycosaminoglycan. So that's the glycosaminoglycan. When it's attached to the protein, its name is proteoglycan.
And it attaches to
the hydroxy amino acid.
Hydroxy.
Yeah, it's a serine or it could be threonine. Yeah, it doesn't attach to other hydroxy amino acids like the tyrosine and so on. So like that. So here are some examples of the repeating units of some common glycosaminoglycans of the extracellular matrix. Here you can see here's the this is the hyaluronic acid. Glucuronic acid and acid glucosamine, right? It's a beta-1,3 link, and then beta-4. And then how many such molecules are the repeating units? It's 50,000 units. Now you can think about the molecular weight. I think next slide you'll have some idea what the molecular weight of the site is. It goes in like hundreds of thousands and millions. So this is the example of hyaluronic acid. This is the chondroitin 4-sulfate. But the chondroitin also could be exist in 6-sulfate. Because the 4 means is the 4 position is sulfate. Okay? 4 position, sorry. The 4 position is sulfate. Or it could be 6 position could be sulfate. Okay? So that they will probably have about 20 to 30 such chains. Keratin sulfate is the only one from this family which does not carry any uric acid. It has amino sugars, an acid glucosamine, and a hexose called galactose. No uric acid. Okay? So this is the only exception here. And then we have a heparin or heparin sulfate. That could be, this is a notorious molecule that they probably can carry 2.5 sulfur molecules per sugar. So you take it to the N-acid glucosamine, right? And it's sulfated. It's 3-position sulfated, 5-position sulfated. And also uronic acid or iuronic acid. Now, how do you differentiate whether this is uronic acid or it's iuronic acid? I means the way it says that ID is iuronic acid. So if you look at the carboxyl group, is towards the up, it's a glucuronic acid. One is down below, is the iuronic acid. Structure doesn't change except the position of the orientation of the group changes that. Now, here are some structures have been repeated. Here, the dominant sulfate body I did not have there. It's iuronic acid and acid galactosamine 4-sulfate. Now look at the molecular mass, the size. Molecular mass, the molecular weight. It's 10 to the power 5 to 10 to the power 7. It's 100,000 to 10 million. It's huge. So each one of them. And so then how do you fit them in a biological system? Do you have any idea? All these heavy duty molecules are extremely important. And they have to be present. They're part of the extracellular matrix and all those. They are involved in many of these biological processes with the normal behavior and also for other pathophysiological conditions. The question is, how do we fit them in all of those structures? They are all present at the same time. They are not being made and degraded. They are not like when you're building a house, you need a cement, right? Kind of thing that they are there. Because they have a certain role to play. So how do you fit them? Does it tell you something? Yeah, but this structure, does it tell you something? And then, like as you say something at home or something like that, It's a kind of not in a biochemical terms. Anyway, the idea is that, can you imagine what is exactly this structure could be look like?
It's not like a membrane.
I'm talking normally, but I don't think, I know what normally we call them, but I'm trying to bring this one in the form of our dentist friends. That's how we can tell it. It's the brush. But it's not the idiom, right? That we normally use it. It's more than the same thing. The bristles are coming out, right? It's like a bottle brush. The baby bottles?
The baby bottle.
Yeah, how they clean the bottle. Because that wasn't available for a long time, right? And hardly anybody said anything about the brush we use it for cleaning our teeth, right? That's because that has a multiple bristles coming out, right? It has a metal thing, at the center, that they wear, and the from there coming. That's like your metal. And what is this one here? That's the hyaluronic acid. Because hyaluronic acid is not attached to any protein. They're pure 100% glycosaminoglycan. And you can see that what is the molecular weight, and how many of those repeating units are present. So, and two that are attached to all the different structures. All the spikes are coming out, the bristles. And it could be androgen sulfate, it could be dermatin sulfate, or it could be hyaluronic, it could be converted 4, converted sulfate, all of those things. And then there, now this segment is taken and it's magnified here. This is your, how they are being linked. Here you have hyaluronic acid. And then you'll see that all this dermatin sulfate, different colors here, and converted sulfate, and they are attached to the protein. Okay, that's called the core protein. And the core protein attaches to the to hyaluronic acid, and it leaves a kind of a not coupling, it's kind of a link, some way to attach that. And that is another type of protein called a link protein. So that's the way all of those things are attached. I mean, I'm not talking about their, it's giving the same structure, like if you're standing upward or lying down, because basically they're lying down with extracellular matrix on which the cells are attached, basically, and then crawl. And without the extracellular matrix, the cells are not going to survive. Those who are attached, those who survive, what's called the adherent cells, not like bacterial cells. They're free floating, right? So have you done any cell culture studies in your life? Anybody culture mammalian cells? Have you seen it? Well, that's what it is, because mammalian cells, you cannot grow them in a broth. They have to be plates, tissue culture plates, you call them. And so that's what the matrix are there. And the moment you lift it down, that's what you do with the trypsinization to remove them, is you break those bonds, and then it becomes available, and you can transfer from one class to another class, or then use it for other purposes. So that's what the extracellular matrix is coming into the picture. So the proteoglycans, they could be soluble, located in extracellular matrix, and they're integral transmembrane protein. And they have many different functions as they can modulate the cell growth. Heparin, heparin sulfate, they inhibit cellular proliferations or internalization of glycosamine glycan. Fibrous growth factors, they do bind to some of them in order for the extracellular signaling to function and bring them the ligand, bring close a ligand to the receptor closer, so the biochemical reaction can start. Whatever the way it is, either for proliferating or promoting the cell growth or inhibiting, say for example, the blood clotting processes. The transporting growth factor, many of you are familiar with that, they seem to stimulate the synthesis and proteoglycans in certain cells. And there are also several proteoglycan proteins, like lymphocyte homing receptors. They have a very similar sequence of epidermal growth factors, which is basically promote the cell growth. And it's been discussed quite a bit, not in normal cells, but also in cancer cells. And they are targeted to develop some sort of therapeutics so that you can reduce the expression of those proteins. So you can, quote unquote, you can slow down the cancer growth, or probably you can try to eliminate it. Okay. Now, hyaluronic acid, as I mentioned, it binds to any cations and in water molecules. And it's present in the connective tissues, in the synovial fluid, and features of the eyes. And it has, because of its chemical nature, it has a different biophysical properties also. The low shear stress, it forms, say, tangle mass and impedes the flow. So it has a viscoelastic properties that should be used as a shock absorbers or as a lubricant, I mentioned before. It's present in where? The cartilage, the umbilical cord is very rich in hyaluronic acid, and that's what it helps during the childbirth. Hyaluronidase, which is an enzyme secreted by some of the pathogenic bacteria, can hydrolyze hyaluronic acid and making tissues more susceptible to bacterial system. This is good or this is bad?
I think it's bad.
It's bad. Now, can you think about it? A good reason for doing this. The same technique for a good reason. I mean, the same approach. Is the bacteria when they infect, they'll try to kill us, right? But if you have the similar approach with hyaluronic acid, or hyaluronic acid is bringing, making of the hyaluronic acid,
I think you can develop an antibiotic against the bacteria that can. It's
no, I
available.
understand that, but I'm talking about another biological phenomenon where this can be used for a positive reason, not for the negative reason. You understand my question or no?
I don't think I understand it.
Okay, this is negative, right?
Yeah, it is.
But what I'm saying, take this example and give it in a positive spin for another biological process. So that becomes it becomes like a positive rather than a negative, which is not a life-threatening.
Well, wait, I'm thinking too much, I think.
No, sure, that-
When you get a bacterial infection, you develop some an immuno. It's immuno-based, the answer you're looking for?
No.
Sorry.
You have to take your mind off from bacterial infection right
Okay,
now.
I'm
So are you
sorry.
saying like, why would we hydrolyze HA?
No, we are bacteria, hydrolyzed. Yeah, right. Well, in our biological system, something does exist, and that HA is hydrolyzed, but for good reason, not for the bad reason. So that's whether anybody knows about it. No? Okay. So I'm going to flip the next slide. Hopefully you'll have the answer. And then you'll say, oh yes, I know it. During fertilization. So this is not bad. You have to make some antibiotic against it to block these processes. Well, it could, but that's not. But the question is, yes, you see that one case is negative, but depending upon how the biology is using the same approach for the good reason and for the bad reason, the same. So the bacteria, they're trying to infect. That's a good reason. That's what they are there for, right? To infect. But they have to survive, so they probably eat whatever they can find. But again, the same thing happened in our human system, animal system. That's here. During the fertilization process, the sperm will hydrolyze an outer GAG coat, and that's what exactly when the ovum, so the sperm to penetrate. So this is during fertilization. So that's a good reason for that. Okay, the other ones is the chondroitin sulfate. It's more similar properties they do have, and it's the strength of cartilage, tendon, ligaments, and the valves of the aorta. That's what they're present. Chondroitin sulfate is mostly you'll find them in the skin, blood vessels, and heart valves. Keratin sulfate is the most heterogeneous GAG with no uronic acid. It says a hexosamine and an hexose. It's present with cornea, cartilage, bone, as well as all this hair, horn, hair, hooves, nails, and claws. All right. So, and the heparin, which is not a constituent of connective tissue, is present in the mast cell, and it type of leukocytes and inhibits the blood clotting process. So that's the normal or natural coagulant. People use it, but it's at the same time it's very expensive, so come up with its different type of molecules, a lot cheaper. But also, the heparin is from the biological sources. You probably come across this I don't know how many years ago it happened. Many of those things, we normally don't make it here. We import it from other countries. And one batch of heparin came from China. Instead of saving lives, it killed people. So there's a huge investigation that what happened to this molecule. And they came up with it, probably they have much more. A molecule is more sulfated. So that means more negatively charged, and so it become more toxic. So that's the ultimate report, what I remember seeing it or hearing about it. Okay, so the proteoglycans, there is several membrane proteins. There's a family of proteins, synthicon, versicon, and all of those. Now you can look into this as your membrane, outer membrane, outside and inside. All the carbohydrate containing molecules, the carbohydrate is always facing outside, not inside the cell, outside. Exothermal domain. So here, with the synthicon, it's about 56 kilodalton protein, like I said about the hyaluronic acid, remember? Yeah. That is the carbohydrate, but this is the protein, and it can be attached to all the GAG chains can be attached, the heparin sulfate, or chondroitin sulfate, and so on, a number of things. Now, if you take the heparin sulfate part, and then you have two different segments, green and little gray area. The green is the S domain, which is sulfate domain, sulfur containing, and then in acetic domain will be the gray area. Now, if you just take that part and you can analyze the type of sugars being present, you will see the same type of sugars as there. The repeating units, the acid glucosamine, glucuronic acid, glucosamine sulfated, iduronate acid, 2-sulfate, 6-sulfate, that notation is still there. So you have to sit down a little bit to understand. They go, this is the guide. You can look at that and you can find the same thing. The repeating unit we've been talking about is there, and the uric acid could be different, but nevertheless, that one has. So what exactly, what type of function it can provide? That's what the bottom line is. So that's the next slide here. This is in the S domain area. So the question is how the heparin will act as an anticoagulant. So now we have several ways, and obviously that it's too early for you to understand how the coagulation cascade. It's complex, but it has to be very rapid. So there is an S domain, the heparin sulfate S domain, that AT stands for antithrombin. Okay, so this is thrombin is a protease, and that's will activate many of those factors of the coagulation cascade. So they are in zymosin, and they're activated, the coagulation starts. Sometimes you have to prevent them, and sometimes you have to do that. So if you have to block the action of antithrombin or factor Xa, so for example, the coagulation cascade. So here is the heparin sulfate. It's here. So when antithrombin binds, there is a conformational change in the molecule, right? It's a square, it becomes this shape, right? Now, the factor, because it will fit exactly to this structural component of the factor 10A, thereby making the factor 10A completely inactive. It's not going to be working anymore. Not going to call it coagulation, so coagulation will be interactive, inhibited that. That's one aspect. The other aspect is also increase the protein-protein interaction. So thrombin here is a different type of molecule, and given an example, it's two different segments. It says the hind leg probably fit into this, and the another domain, the S domain, and the front, the little groove is here will fit the antithrombin, forming it. The whole process will be inhibited. So that's the protein-protein enhanced. Now, biosignaling or extracellular ligands, receptor-ligand interactions, is this matrix. The S domain and heparin sulfate, they will try to bring all of them close together so that they can interact fast enough. So there is the say FGF receptor, fibroblast growth factor receptors, right? And they have these the fibroblast growth factors. So the growth factors is here just to bind to the receptor, and then the biosignaling starts, and then it completes the cell, right? So the heparin sulfate, the extracellular matrix, will try to bring all of them close by so that the interaction will start, will be fast enough to start the signaling process. And sometimes the lipoprotein lipase, when they're going to hydrolyze the molecule, if they are too far away, they cannot work. So they have to bring them closer. All these enzymatic processes to take place. And then again, you see the S domain is helping them to that, so they can hydrolyze. Okay, so that's an example, one of the examples of how it can help us in different ways. So this is proteoglycans, so coming close, right? So the last part is the glycoproteins, which is very, very important. Because basically, as I said, your saliva will have a lot of glycoproteins. The end-length glycoproteins, the proteins are glycosylated. In this case, now what's the difference between proteoglycans and the glycoprotein? In proteoglycans, 95% is carbohydrate, only 5% is protein. In glycoprotein is just the opposite. It's a major part of the protein and very little carbohydrate. It's at a certain length, but it can appear in multiple places of the protein. Protein sequence, I would say, the amino acid sequence what you have. So the oligosaccharides, it could be attached by the N-glycosidic linkage or by the O-glycosidic linkage, but or the bonds. Because O-glycosidic linkage needs threonine or serine, and this is the asparagine residue of the protein molecule. So single protein may have several of these N and O. I'll give an example of the sites available for the oligosaccharides to be attached. Yeah, and they may vary from one cell to another cell, but that's called the glycoform. There's a little micro heterogeneity and variations there, but that's all right. That's not very important there. So glycoproteins have covalently attached carbohydrates. Here are the different way they get attached. Okay. This is the all-linked oligosaccharide, but this is the serine residue where your enzyme galactosamine being attached. Single sugar or to the multiple. This is salicylic acid, and you can have different sugars here. All right. And the N-linked glycoprotein, which is the asparagine residue, which has to be a tripeptide sequence, asparagine, X, serine, and threonine, then only it can be glycosylated. So many proteins are glycogen, you can say they have so many N-glycosylation sites, but not necessarily all of them are occupied. The question is why? We have got no answer at this moment, because the attachment of the sugar and the protein synthesis, it goes hand in hand. So the moment the protein is being synthesized, it's also being conformation, it's changing, folding. So during the folding process, the sites are not accessible, they're not glycosylated. And you cannot glycosylate them in vitro. So that's why many times people try to develop some sort of vaccines, especially for HIV, they fail. That technology, that technology doesn't exist. It can be done too. Because once the protein is folded, in order to get them, you have to bring them as like a straight chain molecule, and you'll destroy the structure completely. So it has to be an active process, an active process. Now this could be, it can give you high mannose type structures, an asparagine to as the glucosamine and so many mannose, or it could give you the hybrid type of structures, or the high mannose molecules, and this is N-acetylglucosamine galactose, and aromatic acid. And this is more complex. It's much easier, the N-linked glycoprotein, because the question is, what you're saying, or one of the things that we say is that if you look at the pentasaccharide, 2 and 3 is 5 sugar molecules, right? This is common, whether it's a high mannose, or it's a hybrid, or it's a complex type. What is changing on this N10? What other sugars are being attached? Now you can tell me that I am telling you all the sugars, and how do I say it so the authentic way that this is the sequence? That this is the sequence. Because after mannose, when you're going to this complex, right? It's going to extend it to the complex molecules, means they are like kind of a heteropolysaccharide, adding more different type of sugars. It has to be an acetylglucosamine. The next sugar has to be galactose, and the terminal has to be salicylic acid. And if there's a fucose, the fucose has to come here. Who decides that? Absolutely, there is no codon for adding those sugars being built. So there's a glycone. It's a hidden code. Nobody has discovered, nobody knows how it is happening. But one thing we know, the structure-function relationship, the enzyme recognizes where it's going to transfer, the acceptor. So that's the way at this moment what we go. If that acceptor is not correct, it's not going to put on the active sugar. Okay? So that's much more complex than what you saw in glycosaminoglycan biosynthesis. Okay, here the example of the glycophorate. It is present in the erythrocyte membrane. It's a transmembrane protein because it goes from the membrane from one side to another. It's a carboxy tunnel, the same terminal. And look at those, all these, there are diamonds, right? The red diamond, there's so many red diamonds, serine, threonine, and all of those. But we have only one blue diamond there. Right? That's an asparagine residue, right? Here it is. So these are all for the O-link, the red ones, the O-link tetrasaccharide. It can have an galactosamine, galactose, and salicylic acid or N-acetyluramic acid with 2 of these. But the blue diamond is the N-linked glycan chains which we're talking about. It must be like a complex type of structure. So in a particular glycoprotein, you may have many sites. Some of them are glycosylated, some of them are not, because they are saying they're the sites. But apparently they have done the analysis and they said that the sugars are being present. Sometimes you have the sites, but they are not being, the sugars are not being added. As I mentioned, that it's not shown here. Basically, when it's is trying to the protein start folding. If the site is not accessible, the enzyme has to transfer. It's not going to recognize that.
So that's why some of them are not.
Not, yeah, that's exactly this. Okay, and then, yeah, because basically that if you take this one and you can digest it, there's a way, and which is the mass spectrometric techniques, which is called the glycomics. For the protein, people use one, and the sugar people use another one. They're called glycomics, and they can come up with a molecular mass. You can separate out the entire glycan chain. There is a enzymatic hydrolysis. Will not destroy any of those. You just take it out from the protein, and then you can analyze those. Then you can assign the structure. So now you can see which are the documents that have all the structures. So the technology has helped us to understand. Otherwise, it would be blue box. It's a blue box, a black box. The properties of mucins. So you can find them in the gastrointestinal secretions, the respirations, the reproductive tracts, and the membranes of various cells. Since these books are written mostly by the biochemists, medical biochemists, or I don't think any books are written from dental biochemistry, so they hardly give any reference to the oral cavity. So it's our responsibility to put it there. And it is important because at one time in my life I did work with the parodic secretions, so the glycoproteins analysis. And the discovery what I made at that time, and that's why I am using this one for therapeutic for understanding the cancer. So you don't have to work with cancer to have a therapeutics developed on that. Because those are excessively regulated process, and the same thing, that process is same everywhere. When people try to use it, the same technology what we developed, what we understood even to treat the congenital disorders of glycosylation as well. So we had a very simple task at that time to find it out, why these patients have problems with the salivary secretions. And especially we looked in the parotid secretions, mostly focused on that. That's what I keep on giving more references into that, because this is not just simple sputum, it's something more in that. And so it also protects the physical barrier on the epithelial surfaces, because we have different cell types. It's involved in cell interactions, and they contain mass certain surface antigens. And submandibular gland actually they secrete mostly the mucins. But the other gland, parotid has a little bit of mucin, but mostly the glycoprotein. So these are informational molecules in the sugar code. Because they have used different things, and lectins that read the sugar code. I'll tell you in a second what it means, and then mediated many biological processes. These lectins came from the plant sources to understand the function of the carbohydrate residues and the glycoproteins, and probably the same thing is happening inside the human body as well. We cannot replicate many things, but we can extrapolate our findings. The lectin carbohydrate interactions are very strong and highly specific. There's not a covalent interaction. It is the affinity. They can recognize some lectin has affinity for some sugars. So you know that what are the ones that in the concatenavalin A, for example, with glutamine, or ricin, and all of those that you find those are plant lectins. I think I have an example here. Yeah, here's the concatenavalin A, which is this. It's loved for the mannose basically. The with glutamine is a WPA, and this recognizes this type of structure. So it's more complex type of molecule, methylglucosamine, cyclic acid, and all those. Ricin is the type of lactose, glucose galactose. Right? The ricin goes for the galactose, so they can think of that. And not only that, the ricin is very extremely dangerous molecule, right? We already have the bioterrorism part of that, because it also blocks the protein synthesis in what we can rely. So that's why it becomes so toxic. Now, many of those infections, the viral infections, so the way the virus interact with the cell surface, they have a cell surface is basically the lectin type of interaction. They have molecule on the cell surface, and the virus or something else, and so their synergy exists, and that way they land it like a landing on the moon, and then they inject DNA or RNA inside the cell and start replicating process. The influenza virus has the agglutinin, which are this type of structures. And cholera toxin, which binds to the it's not a vector, it's a vector single lipid is called the ganglioside GM1, enterotoxin binds here. Let me see, HIV is not here, right? Yeah, HIV, the GP120, we talked about that, remember?